Okay, so what we're going to do is start to get into DNA and talk about DNA structure and DNA replication, and that's really the main thing we're going to get into now. And then we're going to talk about um, genes within the DNA, and then how the genes are expressed, meaning essentially how the information in the genes is actually used by the cell. And then we'll talk about how that information is passed on to other individuals, how it can be modified, and that will lead to how do organisms get different types of information um, so that they could adapt or change over time, which leads to evolution, and which will lead to then diversity within organisms, which leads to um, biodiversity on the planet. And that's kind of like where the course continues to go. Like just actually, so kind of starting with this, as it goes through the rest of the course, what are we eventually covering? We're covering organisms and the diversity of organisms and ecology and evolution and these other things. And it kind of starts off really in a way with the DNA of the cell. So we kind of need to go over this stuff first. Before we get into it, giving you just a little orientation here. So we've had the cell membrane, right, which was a phospholipid bilayer, and we talked about the mitochondria, within the cell. So also within eukaryotic cells, and that's why they're called eukaryotes, right? Eu meaning true, and karyo standing for nucleus, right, or nuclear. Right? So we have a structure that's a membrane that contains the chromosomes of the cell. So we have inside here pieces of DNA, and they're structured into large molecules that are DNA and protein combined that we call chromosomes. And we'll talk more about those as we talk about mitosis and meiosis. So you get a lot more on specific chromosomal terminology and all these things. This is just a quick, broad introduction to it, and then we'll focus on the DNA and then come back to the chromosomes and all later. But a couple things, because we talk about membranes and organelles and all, that I want you to have as sort of a big picture perspective thing. The nucleus is not just a little structure that's just kind of floating around the cell like this. The nucleus is a double membrane structure. So the nucleus is double membrane. So remember, the mitochondria was also a double membrane structure. So this is represented by a phospholipid bilayer here. Then there's a second phospholipid bilayer that goes around the nucleus. Now, I drew those little interrupted areas because the outer layer of the nucleus is actually another organelle in the cell called the endoplasmic reticulum. Well, that didn't quite fit in there, but so sometimes just referred to as the ER, or endoplasmic reticulum. And that structure within the cell is important for a lot of different reasons. One is that's where the membranes are actually made, so it makes membranes. It also is a place where proteins are processed. So the membrane proteins, it's how they get in the membrane, is actually happens here, and then they get transported to the outer part of the cell membrane. All the different enzymes that end up in compartments, like lysosomes and other digestive structures, right, within the cell, right, happens here. It starts to happen in this endoplasmic reticulum. And the information to make the proteins is in the DNA, and then the proteins, before they become functional and actually do their job, need to be processed by the ER. So the endoplasmic reticulum is kind of right there, right with the nucleus, kind of surrounding the nucleus. Things can't even get out really of the nucleus without coming into contact or bumping into or some way interacting with the endoplasmic reticulum. So, and that's because protein synthesis will often take place attached to or on the surface of the endoplasmic reticulum. So I'm kind of putting it out there now because it's one more of our organelles that to talk about within the cells, and it's going to kind of be around, and we'll come back to it here and there. We're not going to focus on it. It's not the main point of what we're doing right now, but just to give you perspective. So what we're focused on at this moment is the DNA molecules, and we're going to start to get into the chemical structure of the DNA that forms the chromosomes that we find inside the nucleus of eukaryotic cells. Right? And the details of the structure are going to be the same for bacteria and prokaryotic cells, except they just don't have a nucleus to contain it. it's free within their cytoplasm. Right? But the DNA that we're getting into now will be the same in bacteria and archaea. So I'll leave this here for a minute, and I'll probably have to erase it later. We're going to look at the DNA. So DNA stands for something. It's not just a thing. So D stands for deoxyribo. And then the N stands for nucleic. And then the A stands for acid. So DNA is really deoxyribonucleic acid. Like, okay, what is that? Right. And that's what we're getting into. So the ribo part here is referring to a sugar, which we've already covered a little bit. And so what I have over here, you will not in this class, the non-majors intro biology class, have to draw the chemical details of this, but I'm going to draw it for you and add to this drawing and show you some extra details just so you have some perspective. And then I'll redraw over here what you really should be able to draw. You should be able to make some basic drawing and representation with labels, and that will demonstrate you actually understand the basics of the structure and how parts of it connect. You do not have to put individual atoms and elements and all into it, except unless there are one or two that are really important, but not the whole thing. So what we have is a sugar. Okay, so a sugar, remember, is made up of carbons, oxygens, and hydrogens. And so the sugar ribo, right, we're talking about here, is really a sugar called ribose, right, which is a 5-carbon sugar, which means there are, remember the formula, right, for a carbohydrate was CH2O with an N, and this would be 5, right? So that would be 5 carbons, 10 hydrogens, 5 oxygens. So that's ribose, the sugar. So what we have here is when you see these ring structures, when you see structures like this, the corners right here are always going to represent a carbon. So there's actually a carbon in them at each of these joints here. So like that's a carbon there, a carbon here, a carbon. Carbon at the end of a line would be a carbon, as well. So these are the carbons of this sugar. Carbon number 1, 2, 3, 4, and 5. There's 5 carbons. So as a rule for sugars, if you remember, every carbon was supposed to have an oxygen. Okay? So these carbons here would have had an oxygen that would have been part of a bond, but that oxygen was lost when it formed a bond here with this to form water. So that oxygen is gone here. This one would have an oxygen. This one would have an oxygen as OH groups. This one's oxygen is here. And again, this one's oxygen would have been lost when it formed a bond here with this guy. So some of them are missing. And they're missing due to the fact that they formed bonds and then the oxygens went away with the bond. Now, there's ribose and there's deoxyribose. So what's deoxyribose? It seems to imply that it's missing an oxygen. So are we talking about these oxygens that aren't quite here? It's like, not those. We're talking about not an oxygen that was just normally sort of shed as part of a condensation reaction to form a bond. We're talking about an oxygen that just should be there, and it's not there. So for example, if this oxygen is just gone, there's no special bond, there's nothing new that's attached here, it's just missing an oxygen. So now this sugar that I have represented here, which is exactly the same as the one I just drew, except I erased an oxygen, that sugar is called deoxyribose. The difference between the sugar ribose and the sugar deoxyribose is here. This number 2 carbon, deoxyribose, is missing its oxygen. It's deoxy. It's without an oxygen at that carbon. And the main reason why that's significant is that's just where the name comes from. Really. That is not going to be part of any connection to anything else or reaction or anything in the future. It's just part of an identifying characteristic. Okay. So how do we know whether we have ribose or deoxyribose? You just look at the number 2 carbon here. I'm going to call it 2' in a second. I'll get to that. And at that 2' carbon, if there's oxygen, it's ribose. If there's no oxygen, it's deoxyribose. That's really the difference, one of the big differences between DNA and RNA. So DNA has the deoxyribose sugar. RNA is going to be almost exactly the same as DNA. It just has the sugar with the oxygen that it's supposed to have. So it's ribose. So it's the actual sugar. So it's called ribonucleic acid, RNA. Okay, so if DNA and RNA are going to contain a sugar that is very, very much the same with just one sort of slight difference. Now, what is this overall structure that I'm drawing here? It's not all DNA or anything. It's actually a structure called a nucleotide. So DNA, though itself, okay, has part of this. It's a polymer. All right, what's a polymer? It's a structure that has subunits that repeat over and over and over and over and over and over again. So sugar attached to a sugar attached to a sugar attached to a sugar, and you just keep going. That's a polymer of sugar. So things like starch, right, and cellulose, and these types of big, large, complex sugars are polymers of simple sugars like glucose repeated over and over again. Proteins, well, they are polymers of amino acids. So they're amino acid attached to amino acid attached to amino acid, and so on until you get a long string of amino acids, and that becomes the polypeptide or a polymer. Here, the individual unit is called a nucleotide. And the nucleotide is a little more complex than these others that are really just follow a formula, like the carbohydrate formula or the amino acid formula. They're very, very similar to each other and very not simple, but they're not super complex. This one's a little more complex because it has really 3 parts to it, and I'm trying to color code the parts here. On this bluish purple part here, the orangey part here, and then the green part there. Okay, so those are 3 parts. So polymer of nucleotides, and a nucleotide is made up of 3 things. There is a 5-carbon sugar, which could be ribose or deoxyribose, 5-carbon sugar, a phosphate functional group. So that's this. So a phosphate functional group is also written as PO4 negative 2. Okay, so it's a phosphorus element and then 4 oxygens surrounding it like that. So one of those oxygens would join to the carbon here of the sugar. One of them is a double bond, doesn't do anything else. These here, they would have been at one point in time OHs, but this is a very powerful acid. If you remember, what do acids do? Acids give something up to a solution. They give up protons, right? Positive charged subatomic particle called a proton. A hydrogen has 1 proton, 1 electron. So if the proton goes away, all that's left is the electron. So these oxygens donate protons, keep a negative charge as a strong acid. They never get that back, so they stay negative. So they're always negatively charged. So phosphates are negatively charged. Keep that. That's an important characteristic of some of these molecules. So phosphate, a sugar, the phosphate is always attached to the number 5 or 5' carbon of that sugar. And then there's something called a nitrogenous base. The nitrogenous base is the thing that you may have heard of as the like A's, T's, G's, and C's of DNA. So there are these molecules. Now, most of our molecules in biology are carbon attached to carbon attached to carbon to carbon. That's the way the lipids were. Fatty acid, carbon, 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 sugar, carbon, 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 other things attach to them. The core is carbon. This structure is nitrogenous, so obviously nitrogen, right, is going to be a big part of it, and that's because Here we have nitrogen attached to a carbon, attached to a nitrogen, attached to a carbon. So there's alternating nitrogen, carbon in a chain. That's unique. Not a lot of biological molecules have that. They're pretty much all carbon core. This one has an alternating nitrogen carbon structure. So that's why it's called a nitrogenous base. You don't have to draw that or anything like that. Usually we'll draw it. I'll just put like, you'll put like N in the middle of a little square, something like that. I'll draw it later for you. But that's the idea. This is a specific one here, just showing you one of the structures. They interact with each other, which we'll get to when we get to the structure. And that's pretty much it. So a nucleotide is a structure made up of a sugar, a phosphate, and a nitrogenous base. That's those are some of the extra details. So some of these details are even kind of beyond what you even need to know, especially the drawing over there. What I'm asking you to know is this. So let's just do we could draw a five-sided little shape like that. I'm sure you can draw a P with a little circle around it, okay? And then a little square with an N attached to it like that. Okay. You can draw that. You should be able to draw that. That's the structure of a nucleotide. And there's a couple other things you need to add to it, and these become really important for later on, right, in the structure. Here is an OH group, okay? This one here, which is the number 3 or the 3' OH group, is very important. Because if you remember, this was, I said the number 5 or 5', and this one is the number 3 or 3'. When talking about DNA replication, and a lot of the processes that we talk about, that in transcription, as well, the terms 3' and 5' get thrown around a lot. So if you read into a book or look up somewhere or look at some of the structures, you're going to see 3' and 5' discussed, labeled, and all these sorts of things. And many people have no idea what that really refers to, but there's these rules that you have to learn related to something reads 3' to 5' or makes something 5' to 3', and you're like, I don't know what these prime things are talking about. Just talking about the carbons and the direction. So it's kind of like saying this end here is the 5' end, and this end here is the 3' end. And when we just look at the 1, it's kind of like, okay, I guess, but it's really more that we said that DNA, right, is a polymer, right? So it's many nucleotides joined together. So if we then start to join nucleotides together and make this chain like this. So every time I'm just doing it, I'm doing a little like 5-sided little shape with the oxygen at the top here. That's the sugar. I'm drawing a little nitrogen, the nitrogenous base, over here, a phosphate sticking off it. So again, it makes it a little bit easier when you see a couple in a row, right? This is the 3' end. This is the 5' end. So sticking off of this end of that piece of now DNA, a polymer of nucleotides joined together is a phosphate. And sticking off of this end down here is an OH, a hydroxyl group, and that's the 3' end. And that becomes very important later on as we add more to the detail of the structure. The ends, which are really the nitrogenous bases, so nitrogenous bases and bases, we'll just call them, would be A, T, G, and C. But they're not just letters, they're molecules like this, and the letters stand for a name. So the name adenine, thymine, guanine, and cytosine are the actual names then of the 4 different molecules that would be represented by different versions of this structure. Okay, so this is just one of those. But they all have some similarities and then differences that make them unique. Okay. So, but we usually just refer to them by their letters, and sometimes even just super generically. We don't even know if it's an A or T or G or C. In this particular one, it's just a nitrogenous base because we're being very, very generic here as we introduce the structure. Okay, so hopefully this we're starting to get a picture. Okay, so DNA is what makes up the information molecules in our chromosomes. The chromosomes are inside the nucleus. They're protein and DNA. The protein organizes the DNA, and the information is held in the DNA. The DNA is a polymer. It's a polymer made up of units. The units are actually a little bit complex. They have three parts: phosphate, sugar, and nitrogenous base. Okay, that's called a nucleotide. To build the polymer, the nucleotides are joined in chains like this that can be incredibly long. Very, very, very long. Thousands and thousands and tens of thousands of long, very long chains over and over again. The structure is essentially repeated: a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar, and that structure repeats and repeats. So it's really, in some ways, very simple, right? And it's just a phosphate's always a phosphate. The sugar is always the same sugar. In DNA, it's the deoxyribose. That's it. So all this part of it here is all exactly the same, the whole entire structure over and over again. The only thing that's different are what are these things? You know, we have an AAA or an ATA or an AGC or a CG. Like the order then of those then is what codes for information. Okay, so that's going to, so what are these is what the genetic code is going to be. And we put it together. But this isn't quite the end of it. That's not really a full DNA structure. DNA, it was found out to be a double-stranded structure. So instead of a single strand like this, there are two strands that are then held together, separate from one another. So not one strand folded back on itself, but two separate independent strands that are then held together. So one strand here, and then I'll do a different color just so you see the different one. Another strand here. Strands are then bound to each other with some kind of bond, okay? And it's going to be, eventually we'll get to there, a hydrogen bond. So hydrogen bonds are what hold together two strands of DNA. As scientists start to figure out the structure of DNA, and they're like, what, how is it all put together? They didn't know if it was double-stranded, single-stranded, triple-stranded, and we actually do have real-world examples of all those that actually exist. But most all DNA in the nucleus of the cells is going to be double-stranded. That was found out by a woman named Rosalind Franklin. She did a technique called X-ray crystallography, and she determined, based on the data that she received from her work, that there were just two, not one and not more than one, or not more than two, not three of them, but two strands of DNA. And so people started to say, okay, so how do those two strands attach to each other? So there were ideas, things like, okay, so maybe in this strand here, we have an A, maybe a T, a G, and a C. I'll just go like that. They said, hey, does it work like this? Do the A's and A's and the T's and T's and the G's and C's go together? And they found, no, it doesn't work like that. So they couldn't quite fit them together. Years before, a scientist named Chargaff, he looked at DNA before anybody even knew it was double-stranded. And he was kind of looking at A's and T's and G's and C's, and his idea was that they'd all be the same. That A would equal T, would equal G, would equal C. So essentially 25% of each. 25%, 25%, 25%, all the same. And what he found was something interesting. What he found was that this was partially true. He found that A would always equal T, the amount, whatever it happens to be. And G would always equal C, whatever it happens to be. But A would not equal G, and A would not equal C, and C would not equal, and so there was something going on. So something between G and C was unique, something between A and T was unique, and different organisms, he found different amounts of these. So that wasn't even the same between all living things, all plants and animals and different organisms he looked at, he found different ratios, different proportions, but and this was always holding true, but didn't know what it meant. So then, as people started to find out, oh, it's double. I'll leave it kind of there for a bit. As it's double-stranded, they said maybe that's the clue. Maybe that's the key. It's A and T like this, and C and G, and that makes sense with other information. But people, they couldn't quite get it to fit together. So by fitting, and I mean they really just made three-dimensional models, like Tinker Toy models type things, with little sticks and balls, and they kind of got the shape of these things, and they try to put them together to see how they would fit, and they had other data obtained of how it's supposed to look when it's all fitted together, and they really couldn't make it work. So these two scientists, who are the ones who end up being the most well-known in terms of the structure of DNA, not that they found out the most, it's just that they revealed this final point. So Watson and Crick, they found that when you put the two strands together, that A and T will match up and G and C will match up, but they only match up if one strand is upside down by comparison to the other. So meaning, and this is where these numbers come in, if this strand here had its 5' end here, that's the phosphate, right? This one here would have its OH, 3' end. They're kind of opposite. So here's the phosphate down here for this strand, and this is the OH, the 3' for this strand. So the strands are running opposite one another, or the term for that is anti-parallel. So DNA is double-stranded. When DNA comes together, certain bases, certain nitrogenous bases pair up. We always have A pairing up with T, and we always have G pairing up with C. And for the two strands to fit together properly, they have to be anti-parallel to one another, so one has to flip upside down compared to the other one. So, as far as what do you need to know? Terms, basic terms, and definitions of this. You need to know we'll talk more about chromosomes later, but what is DNA? So DNA is a polymer made up of nucleotides. It's double-stranded. There are two strands of it. Each individual strand is made up of a series of nucleotides joined together. The name for the type of bond that joins them together, which, because this will come up here and there, is called a phospho, because obviously there's a phosphate there, and then diester, which is more a chemistry term, as a specific type of bond. And that's just in comparison to the peptide bond between amino acids, a glycosidic link between sugars. So the names of bonds between different molecules have different names, right? And this one has its own special name before of its special properties. Okay, so phosphide ester bonds are the type of covalent bond that links together nucleotide to nucleotide to nucleotide in a strand. But then the two strands are joined together with hydrogen bonds between A's and T's and G's and C's. That's the base pairing. But they only pair up properly if their orientation is upside down compared to one another, or what we call anti-parallel. Which means that if we get to more technical terms, one strand is running 5' to 3', the other is going 3' to 5', right? If we're following them in the same direction, 5 to 3, and this one's 3 to 5 over here. That all those little details will become important as we move on to the next step. So the next thing we're going to do is talk about before cells can divide, before one cell divides into two cells, it has to double the DNA. We need twice as much DNA, so each cell gets the same amount of DNA. So we have a process called DNA replication. The process of DNA replication is highly influenced by the structure itself. And to understand that process, you need to first have some basic understanding of the structure. Because there are certain rules to that process, and there's certain problems and conditions that will happen, and they're all reflected by these structural terms and structural situations. Okay, so that's the end of this for the basic structure. And the next thing we'll go into is the replication process. How do we build more DNA?